resume recording. And okay, we'll just wait a little bit for people to come in. Okay, great. I'm going to go ahead and get started. I know some people are still entering the room. But in the interest of keeping everything on time, respecting everyone's time, I wanted to get started with our NYLCA Lunch and Learn regarding the very important subject of COVID-19 and human milk. We're happy to have you all here with us and learning with us today. I'd like to welcome the members of NYLCA and our board members who are joining us today. And in just a moment, I'll introduce our president, Crystal Alexander, who will be introducing our speaker. But before that, I'd like to take a moment to humbly recognize and acknowledge that where we are in New York City is a part of the unceded land of the Munsee Lenape people, along with other indigenous communities. We understand that the original stewards of this land had multi-generational traditions of how to feed their infants, raise their families, gather together with their communities. And our intention of this recognition is part of the greater work that must be done to dismantle the ongoing legacies of settler colonialism. The New York Lactation Consultant Association, NYLCA, is a professional organization comprised of international board-certified lactation consultants and associate members who are also lactation professionals and a part of our community. Our goal as members is to organize, discuss, and advocate for issues regarding to breastfeeding and chest feeding, lactation support, and promoting the IBCLC as the gold standard in lactation care. NYLCA has set up the free referral line where families can access IBCLC support ASAP, and it's been a wonderfully successful way to get care to families who need it as soon as possible. At the end of this program, you'll be connected to a survey, and at the end of that survey, you'll receive information on how you can help connect families to the NYLCA free referral line and help them get support as soon as possible. We encourage people, if you have questions during this presentation, to utilize the Q&A part of their Zoom. Go ahead and pop your question in the Q&A and I will read them and we can discuss with Dr. Powell at the end. But we ask that everyone remain muted and respectful of our presenter. So now I'd like to introduce our esteemed president, Crystal Alexander, who will tell you about our speaker and the topic today. Go ahead, Crystal.
Hi, Kate. Thank you so much. Happy New Year to everyone, and thank you all for joining us. Thank you, Kate, for putting together this wonderful education program, as you always do. I'm super excited to hear Dr. Powell speak. Just at this time with COVID-19 being such a big part of our lives, and just the impact of it with breastfeeding. I'm just looking forward to hearing everything she has to say. So without further ado, Dr. Rebecca Powell, PhD, CLC, is an assistant professor in the Department of Medicine, Division of Infectious Diseases at the Icahn School of Medicine right here at Mount Sinai. She received her PhD in microbiology from the Sackler Institute, New York University School of Medicine, and her CLC from the Healthy Children Project. Her doctoral research focused on the genomic analysis of HIV in the epicenter of the HIV pandemic, which was in West Central Africa. Her early postdoctoral research was performed at the International AIDS Vaccine Initiative Design and Development Lab, where she was involved in design and testing of replicating viral vector against HIV. Dr. Powell's background in HIV research brought her to join a team of HIV researchers at Mount Sinai, where she first found her niche studying the impact of cellular immune response in milk on mitigating mother-to-child transmission of HIV via breastfeeding. The overarching goals of Dr. Powell's research program are to understand the human milk immune response to infection and vaccination, and to design vaccines aimed to enhance this response. Dr. Powell currently has several active research projects aimed to fill knowledge gaps about human milk immunology, exploring the antibody response to SARS-CoV-2 infection to determine its potential to protect breastfeeding babies, and as COVID-19 therapeutic, the antibody response to influenza vaccination, and the design of maternal vaccines to eliminate mother-to-child transmission of the HIV via breastfeeding. Dr. Powell lives in Brooklyn with her partner and her 3 children. And thank you so much for doing this for us today. We look forward to hearing everything from you.
Thank you for that introduction. Okay, so I'm going to share my screen right now. And I assume everyone can see that. Alrighty. So yeah, thank you for inviting me today. I love talking to all kinds of professionals about milk science and milk immunology. And today I'm going to talk to you about all the work we've been doing, especially in New York City, since our epidemic started here in, March, essentially. So I'm going to talk to you about our evidence of a strong and specific antibody response against SARS-CoV-2 in human milk. And I know we have a lot of different people listening in today, so I'm going to give a little bit more of a background to certain aspects here so everybody can understand what I'm talking about. So many of you are familiar with antibodies. Antibodies are protective proteins that are produced by the immune system in response to the presence of a foreign substance. And this is cartoon diagram down here, the different areas of the antibody. You have the side of it that sticks to the pathogen, and you have the side of it called the constant region that might interact with cells of the immune system, for example. They recognize and they latch onto the foreign substance in order to remove them from the body. And when the alien substance, like a virus, enters the body, the immune system is able to recognize it as foreign because its proteins are very different from those found in the body. But not all antibodies are created equal. Of course, it's very complicated. There are many different types and characteristics of the antibodies in humans and in all creatures. So today, I'm mostly going to be talking to you about IgA, which comprises about 90% of milk antibody. But it's important to understand, and I'll touch on, there are many other classes of antibody, and some of them, most of them really are found in milk in different quantities. And most of you are probably most familiar with IgG, which is what's dominant in our blood. There's many classes of IgG, they perform different functions, and that's probably the most studied antibody. There's also IgM, which is produced early after infection typically. It forms these higher order complexes as shown here, and it's also found in milk. So again, I'm going to focus on IgA, but we shouldn't forget about the other classes because they may be performing important functions. So as I said, human milk antibody is about 90% IgA. And it's also about 8% IgM, and most of that in humans is in what's called secretory form, as I'll show SIgA or SIgM. I'll focus on SIgA today. So these are what's called polymeric antibodies, meaning that it's not just the antibody molecule, but they're complexed or attached to another protein called a J chain that holds together 2 of the antibodies in the case of IgA to form what's called a dimer in most cases. And as well, that whole complex is wrapped in another protein called secretory component. So this again is given in a bit of a cartoon diagram here. There's 2 kinds of IgA. I'm not going to really differentiate today. They do just form regular monomers in blood, and you can find them in blood. But then as a dimer, that's what you find in your secretory areas. So milk, and also saliva, nasal secretions, vaginal secretions. You would find dimeric IgA, and it would be mostly in secretory form. So wrapped up in this really protective additional protein called secretory component. And so that secretory component is essential for the protection of antibodies against degradation in harsh mucosal environments like the infant mouth or gut. So it's very, very important to protecting the antibody and having it be able to act and not get degraded. So in humans, milk IgG, which is about 2% of the total antibody in milk, will come mostly from the blood. It will just kind of seep in. There might be some local production in the lymph nodes near the breast. The B cells that make the milk secretory IgA and secretory IgM to a lesser extent come from what's called the mucosal associated lymphoid tissue, or the MALT. But most of that is actually coming from what's called the GALT, the gut associated lymphoid tissue, with a bit of respiratory tissue involvement as well. So this is called the enteromammary link. And it makes a lot of sense evolutionarily because, you know, the mother is ingesting pathogen essentially, and the baby needs to be protected against those pathogens, and those are the pathogens that will kill the baby, and that still readily kill babies in low-income countries where they are truly dependent on the protection from the mother's milk to save them from such diseases. And so when the mother ingests the pathogen, that pathogen goes to the gut, right? And that there's a lot of immune tissue, specialized immune tissue called the GALT in the gut, in the small intestine, and that generates this very specific B cell response. And those B cells are programmed to travel to the mammary tissue as part of their function. And there's a lot of evidence we have in humans and other mammals for this. And then they produce the antibodies in the mammary tissue that will end up in the milk, be secreted into the milk. And this is really makes a lot of sense, right? To protect the baby. So there are many functions of antibodies, and I'm not going to talk about most of them today, but you can look at this diagram and understand how complex it is and how many different functions antibodies are performing. What I'm going to focus on today are the blocking functions of antibodies and the functions that are mediated by what's called the Fab, or the variable region of the antibody. So again, in that Y-shaped structure that I showed you before that you're probably familiar with, those two little arms are different in every kind of antibody, and they are specific for whatever pathogen you're interested in. And in this case, we're talking about SARS-CoV-2, right? And particularly, we're talking about the spike protein, which I'll get into in a minute, because that's what attaches to the cells. So you see, you have to measure the ability to bind of that antibody to the virus, and you can also measure the ability to truly block infection, and that's all essentially mediated by what's called the variable region, or the Fab region of the antibody. There are other antibody functions that I'm not going to talk about today, but are likely also playing a role in protecting everybody once antibodies are formed against SARS-CoV-2. And those functions are mediated by the other side of the antibody. And as I mentioned before, the constant region of the antibody will interact with cells of the immune system, and they will cause those cells to function as they are intended to do, to destroy infected cells and viruses themselves. But again, I'm only going to really talk about the blocking function of the variable region of the antibodies today that I've studied. So SARS-CoV-2, that's the virus that causes COVID-19. We're all very, very familiar with it. Unfortunately, at this point, I think even just a layperson is way more familiar with this virus than they've ever been with any other virus in their lives. So the spike protein is the protein responsible for allowing the virus to attach to and fuse with the membrane, the outside of a host cell. The spike protein has a very strong affinity for a protein on human cells called ACE2, which maybe you've heard of, and that's what really mediates the attachment to the cell, as diagrammed here at the bottom. So this receptor, ACE2, is on many different cells in our bodies, and that is the reason why infection manifests in so many different ways. And across, the human population, there's probably a lot of differences in how this is expressed on our different cells. So that's why different people also have different manifestations. And there's a lot about that we don't understand yet. So the spike is a very, very important protein. That's the main target for what we would call functional antibody binding. So we make antibodies against lots of different proteins that comprise the virus, right? So that's shown at the bottom here. Many of the tests that look for antibodies actually, as a diagnostic tool to tell you if you've been exposed in the past, they may actually be looking for antibodies against nucleocapsid. This is the N protein listed down here, because those antibodies are formed in a very plentiful way, but they're not necessarily functionally important. They can tell you you've been infected, but they may not necessarily be the ones blocking infection the way the antibodies against spike are the ones that truly are going to be blocking infection. So that's why we focus on those antibodies, why I focus on those here. So a little bit of background about COVID-19 in babies and young children. As you probably know, thankfully, COVID-19 pathology is in children, young children especially, is typically much more mild compared to adults. But about 10% of babies, babies under 1 who contract the virus will experience a relatively severe illness that really, for them to recover, and essentially all of them, mostly all of them will recover, but they require advanced care to recover. And children are likely responsible for a lot of the spread of SARS-CoV-2 globally, even if they don't manifest a lot of symptoms. And a minority of children, as I'm sure you're aware, we've found now are experiencing what's called a multi-system inflammatory syndrome after their infection with SARS-CoV-2. So this can be fatal in certain cases. It certainly causes a lot of pathology, lands many children in the hospital. It's still a very small minority, but because we're in this pandemic, that number is not insignificant. And so for all of these reasons, protecting babies and young children from infection is, of course, essential. And one way to protect babies and young children could be what's called passive immunity via milk feeding from a previously infected lactating person. So this could be the child's parent who is breastfeeding, chest feeding, or this could be from donor milk as well, right? So the milk immune response to SARS-CoV-2, of course, it's a new virus, relatively speaking, and so there's nothing known about this. There was nothing known about this until we started our research, and others are also researching this topic. And research, is urgently needed to test human milk for SARS-CoV-2 antibodies and the function of those antibodies. Understanding the human milk immune response to COVID-19 really is essential at every level for parents, of course, to trust that breastfeeding is the right thing to do, for lactation professionals, such as many of yourselves today, because many are asking us for advice every day. For healthcare professionals, doctors and nurses who unfortunately tend to not, always give the most well-researched answer about lactation when people come to them, unfortunately, and many of them essentially had said, just pump and dump, just be separated from your baby and so forth. And that's still happening today, despite the research. And of course, for scientists, science, obviously, it has to inform public health rather than hearsay and myth and everything that often surrounds lactation, but especially in the midst of this pandemic, we need this research. And, a big part of my research, something that I find very intriguing, that is a novel concept, because milk is really understudied in general in immunology and in many other ways, that the antibodies that if they're produced at high levels, what we call high titers, after infection in milk, these antibodies may be highly durable because they would, one would expect, be in the secretory form that many antibodies are in milk. And they're meant to survive in harsh mucosal environments, as I mentioned before. So I always felt from the beginning that these antibodies could have potential as a COVID-19 therapeutic, because they're very special if we were to extract them from milk, and they're very different from what's in blood. So it's the idea of convalescent serum, convalescent plasma, and blood donation after you've been infected, but it's also very different, and it might be very unique as a therapy. So towards this aim of studying the milk immune response to COVID-19, we've recruited over 800 lactating participants who have recovered from COVID-19. So we have a big New York City enrollment, and it's also a national enrollment. So many of you might have seen our ad. We use social media to recruit. And when this went out in very early April, I worked about as quickly as I could to get research approval. We, posted our usual posts, and the excitement that surrounded that post was enormous. So the whole recruitment went viral, so to speak, and we had such enormous interest immediately, which was great and also very overwhelming for me. And it preceded any money we got to actually study this. So we did finally get funding to do this, but we did a lot of the recruitment and enrollment and sample collection before we really had the money officially to do it. So you probably saw, this viral response to our recruitment. It made it into the news right away, and it all only fed into the continued enrollment. So, it was amazing, really amazing. And it's really continued the interest in this research, which has been great for us. And it's very important because it's brought recognition to milk research in a way that it's really been lacking. So I'm going to get right into what we have found, and I'm going to spend some time slowly going through the data, because I know many of you probably aren't as familiar looking at data as I am, and I don't want to take that for granted. So I'm going to show you data on a few different. We have our pilot study set, which was 15 samples, which is now a published paper. So most of the data comes from that. We additionally have looked at another, at this point, probably 50 or so samples, and many of which are represented here. And everything I'm going to say has held true along the way as we've looked at more and more samples. So these study samples from our various early study, our pilot study that led to the early research that got us funded, thankfully, these were very early samples collected all locally by me, which was a lot of work early in the pandemic here in New York City. So these samples were all essentially collected in April of 2020 when the city was on pause. And so nobody else was really on the train when I was going around collecting these samples, and they were collected, during, I think, the height of our worry and paranoia, which was, well-founded. So they were mostly collected in a very no contact or low contact way. So about 1 ounce, so 30 mils of milk was obtained from participants, and they used electric or manual pumps, whatever they were most comfortable with. Everyone pumped in their homes, continues to pump in their homes, freeze their label in bags, freeze their samples at home. And that lends itself to being easy during these pandemic times, because nobody wants to be coming in anywhere or going out if they're not comfortable with that. So that's worked out well. All the participants I'll show you had a PCR confirmed SARS-CoV-2 infection. Milk was obtained about a month or so, usually between 3 to 6 weeks really after infection. This is just a clip. There's a video from Business Insider of me going around, and I think this is in May actually, or even maybe by June, collecting some of these samples. And so you can see, this was someone who actually handed it out their door. Some people left it on their apartment, stoop. what have you. So this is essentially how the samples were collected. Now, since these very early samples, many of these early participants, I've obtained, repeat samples. This is a longitudinal study, and I'm going to show you some of the data of the longitudinal aspect of how the antibodies have changed or not changed over time. And then we also have control samples, which are samples we already had in our freezers from other studies that were all pumped before December 2019. So it's of course important to have negative controls that can't possibly be positive for these types of antibodies. So I'm going to spend a minute explaining how we test milk for SARS-CoV-2 antibodies by something called an ELISA. So ELISA stands for enzyme-linked immunosorbent assay. So this is a plate-based assay. So it's not really like a dinner plate. It's a plastic rectangular flat plate that has separate little areas for different samples. And it's a technique that's designed for detecting and quantifying soluble substances like antibodies in milk specific for viral proteins. So you can use ELISA to look for the total amount of antibody in a fluid like milk or blood, or you can look for specific antibody in that fluid like specific antibodies against SARS-CoV-2. So the way this is done is, imagine this is the little spot on a plastic plate that's coated so that it sticks to proteins. So you add your protein of interest. So in this case, we have a lab at Sinai that generously has given us spike protein that they have made for their tests. And so we essentially, that gets glued down to the plastic plate, and then you add on the milk that contains what's called the primary antibody. So these are antibodies, if they're there, against SARS-CoV-2 that are in the milk. We use milk. The way we use milk is we get it, we thaw it, and we spin it in a centrifuge so that the cells that we don't use in this experiment go to the bottom and the fat goes to the top. We remove the fat, we don't use that, and we just use the liquid portion of the milk. We use the milk as is at that point, or we dilute it, as I'll show you. And so after the milk has incubated with these plates that are coated with spike protein, you can add on specialized antibodies that companies make that are made to recognize human antibodies. And so then that allows you, because they have a fluorescent tag, to be detected by a machine that detects fluorescence and tell you if the milk had antibodies against SARS-CoV-2. And this is not something I invented. This is an extremely old standardized technique, but we have optimized this technique over time for use with milk, which is not blood, right? So you need to do some different kinds of optimizations in your experiment to make it work with milk. So now I'm going to go through the data from our pilot studies. So what you're looking at here is the SARS-CoV-2 spike-specific IgA in undiluted milk. And this is from 20 people who recovered from COVID-19. That's the solid lines. And then you're comparing it to, I think I have 10 controls here. So these are our pre-pandemic samples. So these samples are our negative controls, people who absolutely did not give this milk during the pandemic. Any light we see in the ELISA, any reactivity, is what we consider background. And so to be positive, these samples have to be above that background. That background level is calculated as the average value of these samples plus 2 times the deviation of that average. So you really, once you get above that pretty stringent cutoff, you consider these samples positive. And if they're not above that, they're negative. So we know these people had SARS-CoV-2 infection. These samples were collected, as I said, about a month after their infection. And what you can see here is that, well, you can see there's a lot of variability in the strength of the antibody response. This is, again, IgA only. It was evident that 19 of these 20 samples from the COVID-19 recovered donors had spike-specific IgA binding that was significantly above the pre-pandemic controls. So this is every single one except one of them shown here, right? It varies, right? And this is just one sample. It's a snapshot, and the immune system changes in different ways over time. But you can see that almost all of them, except one, had the spike-specific IgA. And then what you're seeing here is the kind of data you get when you dilute the milk. So as I said, sometimes we test the milk as is after we've removed the fat. And then sometimes we serially dilute that sample. That means that you would continue to dilute the milk, in this case, fourfold, essentially with saline, and test all those dilutions by ELISA. And why do we do this? Well, because it can give us an indication of the true strength of the antibody response. So it's very common when you use this technique to dilute the samples, whatever they are, if you're testing blood or some other fluid or milk, to really distinguish, sample from sample and get a sense of the true variability. So you can see here, again, you can see that there are samples that are quite potent. You can see that there are samples somewhere in the middle of their potency. You can see that there are samples that essentially, that one that truly fell into the negative, which was this one, right? So again, this is undiluted, these dots. And then each point after that are the serial dilutions all the way out to a dilution of 1 in 1,000, essentially. Now, by the time you get to 1 in 1,000, of course, everything is negative. And you can compare to, the negative control points at 1 in 1,000 or somewhere hiding down here, but they're no different. And you can certainly see that we have the very strong responders, which, I certainly tried to focus on those for later experiments to get a sense of, what the best of the best we're doing. And then, of course, we'll analyze all of these the same way eventually. So one, this, what I'm about to show you, which feeds off of that early data, is very, very, very recent data that I've literally just put together, that looks at many of these samples and also some that aren't on that graph. But as I said, we've actually looked at more samples than what's shown there from our pilot data over time. We have repeat samples because all of these participants are asked to continue to save an ounce of milk every month. And so we have continued to collect locally from many of these people who have been able to stay in the study, and people stay in the study until they no longer can due to weaning or other reasons. And then some people nationally have collected a bunch of milk and have been waiting for us to be ready to pick it up. So we use Milk Stork, which is a milk shipping company that sends out a box that keeps the milk at the right temperature. And that's, we organize that, and people put all their milk samples in a box and it comes to us. So in that case, we get all of the samples at once, and we will test essentially starting with the first and the last. And then over time, we'll look at the interim samples as well. So that's how we get these longitudinal samples. So all of these that I'll show you were obtained between 5 and about 9 months. So this is a while between the samples, And so this is again looking at IgA against spike, and this is really good news, right? So what this is showing you, it's a little dizzying, but just for example, this sample from this person with the number COV for COVID 104, right? So we letter the samples that we get monthly, and so this person, it's about 6 months apart, and you can see that their antibody levels, I'm going to call this unchanged. It might have gone up a little bit. We would have to reproduce this to really demonstrate it's gone up. It does look like it's gone up a little bit, which is possible. And then this next person, similarly, not changed over time. Maybe this is the only one that perhaps has decreased, but again, that would have to be reproduced and look at another sample in a month and see if that's really true, or, just to be sure. But the rest of them pretty much are unchanged. Maybe they go up a little bit. And that is really great news. So that means that, if you had COVID-19 and then You want to know, okay, I had it 6 months ago. Is there a chance that the antibodies in my milk might still be protecting my baby? The answer is yeah. I mean, from what I can see, of course, we aren't directly testing that yet of is it absolutely protective for babies, but the antibodies are there, and they're not changing over time, really, which is amazing. It's very promising, right? And that means as a therapeutic, if we were to do that, we could have a donor pool that is people who were infected 6 months ago, 9 months ago, not just people who were only infected in the last month. It means like if you were ever infected, essentially. So this is great. This is really just very, very, very new data. So, the other thing to keep in mind is that we're measuring IgA, but we don't know if it's secretory IgA until we test for that. So what secretory IgA, as you remember, is that really common type of antibody in milk most of the time. But this is a new virus. We don't know what's going to happen in the milk. And we want to see that most of it should be secretory form, because that's the form that's really going to be durable in the baby's mouth and gut. And also, if we were to extract for therapeutic, that's what we're really interested in, right? So this data looks a lot like the other data set that I showed you, looking at the level of antibody binding against spike, but here we're looking at secretory antibody. Okay, so you can actually see that again, all but one are positive for secretory antibody binding against spike. And again, we're comparing back to these pre-pandemic negative controls. And I always like to highlight at this point, yes, this is looking at undiluted milk, which for some of you that are familiar with ELISA and familiar with how we test antibodies in blood, you would never use undiluted blood plasma for this experiment, because you can't. It's just, if you have your controls, the background is so high, you would never do that. You would always dilute blood plasma for this experiment. Milk is different. You can use milk undiluted. The controls are manageable. They don't give you background that's too high. And in fact, it's very useful to look at undiluted milk for how much antibody is there, to me, because that is highly relevant to the protective effect in a breastfeeding context, right? So this is literally what the baby's being exposed to multiple times a day. This is not diluted, right? So again, now looking at the diluted milk, looking against secretory antibody against spike, again, you see there's a lot of human variation. That's very normal, especially in milk. You get a lot of variation, and you can see, the really high responders, the mid-level responders, the lower responders. We don't know what this means, but it's good to take a look, and then you can later try and dissect what it means to have the stronger response, the mid-level response. Is it relevant or not? Is it more protective or not? We don't know yet. So, just so you realize that, when we measure IgA against spike, and we measure a secretory component of antibodies against spike, we actually don't know for sure that the secretory component that we're measuring is necessarily secretory component on IgA, because secretory component can also be on IgM, and it could be floating free in the milk, although it's extremely unlikely we would be detecting that because of the way that you do an ELISA. But it's always good to try and figure out if what you're measuring is truly relevant to what you're trying to understand. So is this secretory component on the IgA? Now, we know that the data sets looked really, really similar. So, that was very promising. And what we can do is we can measure a correlation between the two different types of data. So when we try and correlate the values between the spike-specific IgA and the secretory antibody activity in the undiluted milk, you can see that just like those data sets visually looked familiar or similar, you can see that the correlation, what we found, because everything falls along a very linear straight line here, essentially, that it was a very, very statistically significant positive correlation between our IgA data of antibodies that bound to spike, and our secretory antibody data, meaning that it was extremely likely that most or all of that IgA was secretory IgA. And again, that's really, really important because we are interested in the true function of these antibodies. So in the baby's mouth, in the baby's gut, in the, the whole oral nasal cavity that's involved when breastfeeding is occurring. I'm sure we have all seen a baby breastfeeding. There's going to be the bathing of the oral and really nasal cavity with this milk, but it's a relatively harsh mucosal environment. So you want to make sure that that antibody is going to survive the natural enzymes that are in those areas, just in terms of the baby. So, what this is showing us is that indeed most or all of these antibodies that we're detecting are secretory form antibodies, secretory IgA, and that's what's going to have that potential to be highly protective for the baby. And then additionally, for the therapeutic aspect, if you were to extract these antibodies, which we can do fairly easily, you want to know that what you're going to get of antibodies that are specific for spike are mostly in secretory form, because those are what are special compared to what's in blood to use therapeutically. We would use them as a respiratory therapeutic, maybe a nasal spray. And we're getting to that point. We're going to start small animal experiments, really looking at that question, because we could target the therapy of these antibodies because they are designed to be in such mucosal environments as the upper respiratory tract. So we have found, and again, this is early data, that most or all of these spike-specific antibodies are actually in secretory form, which is really good. So I'm going to throw a little more data at you to get more of a comprehensive picture of the milk response. So this is data in our paper that came out, I guess it was late November. So we looked at a subset of these samples, specifically looking at reactivity, not just against the whole spike, which is diagrammed down here at the bottom. It's like a little flower. It's actually, spike is actually 3 spike proteins called a trimer that come together in a particular form. And that form, that structure, is very important for how it binds to the cells. But what's called the receptor binding domain is specifically that little part on each of those proteins that ACE2 binds to. So it's a really important target for antibody binding and for what's called neutralization. So how the antibody would neutralize the virus, which means block it from infecting a cell. So it's always good to get a sense of the antibody reactivity against the receptor binding domain. Now, that doesn't mean that antibodies against spike that are not against the receptor binding domain are not important, because there are other ways that these antibodies would be neutralizing the virus, even if they're not binding to the receptor binding domain. But everybody's interested in what the reactivity is against RBD. So we did measure that for a subset of samples. And what we found, as you can see here again, this is diluting the milk and comparing to negative control pre-pandemic samples. Those are the dotted lines. That 80% of the samples we tested, and this is a bit of a small sample set, but 80% of the samples we tested did indeed have receptor binding domain specific milk IgA, which is certainly important to note. And then beyond that, we also looked at the IgG response or the IgM response against RBD in this subset of samples. What we found is that the IgG and IgM response was certainly not necessarily concurrent or measurable when you have a IgA response. So the IgA response was dominant, and sometimes we found IgG, sometimes we found IgM. Occasionally we found both along with the IgA, and this again looking at 15 different people who were infected with COVID-19. But the dominant profile was that they just had IgA. So that was interesting and not necessarily surprising, because total milk antibody is comprised of about 90% IgA, right? We were a little bit surprised we didn't detect more IgM, but that could have been in terms of the kinetics of when we were measuring it. Because if that IgM was earlier, we would not have measured it, 4 to 6 weeks out. The IgG is in such a minority in the milk, it is also possible that our assay, our test was not as sensitive, and was not sensitive enough to detect whatever IgG was there. But for all intents and purposes, if that was not detectable by our experiment, then what effect would it really be having for the baby in undiluted milk? I would hypothesize very little, even if it was there in quantities we could not measure. So this is summary data looking, at the whole antibody response from those single samples. So again, what's happening here is that you're getting SARS-CoV-2 infection. That is causing stimulation of the mucosal lymphatic system, the mucosal B cells. So there might be some respiratory mucosal involvement. There's certainly going to be gut mucosal involvement. And then those B cells really probably very quickly transit to the mammary gland, as they are programmed to do, because that is how we are evolutionarily wired. And then the IgA, primarily and dominantly IgA, is secreted into the milk as secretory IgA. And I didn't get into this in detail, but part of the process of that antibody being secreted is actually, as it goes through the cell membrane from the mammary gland into the milk, it gets wrapped in that secretory component. So that's actually when that happens. And that secretory component is very protective. So about 4 to 6 weeks after infection, what we found was that 95% of the milk samples we looked at contained spike-specific IgA. And then about 80% of those were specific for the receptor binding domain. And then that means about 20% would have been specific for other domains of spike. And we don't know which domains right now. And then of the 80% that were specific that had RBD specific IgA, there was about, as I touched on before, about a third of those just had IgA. About a third of them had IgA and detectable IgG and detectable IgM. And then you can see about a quarter had IgA and detectable IgG, and then a very small minority had detectable IgA with detectable IgM. So we don't really know what this means, and it needs to be studied much further. But we know that the response is secretory IgA dominant, and that is the class that we were most interested in detecting for various reasons, as I've gone into already. So the last bit of data I'm going to show you is looking at measuring neutralization activity. So I mentioned before, so antibodies can neutralize a virus. What that means is they can block a virus from infecting cells. And they do this using the two arms of the Y shape of the antibody that are what's called the variable region, that are specific for that virus. And if they can bind in a certain way, they literally stop the binding of the virus to the cells. So they neutralize it. It cannot enter the cells. So neutralizing antibodies are very important for SARS-CoV-2. It's what everybody is looking at and interested in the blood after infection, and certainly what everybody is also very interested in after vaccination, because we really need a lot of antibodies. They're going to just stop that virus from infecting cells. So the way that you can measure neutralizing activity, you can use real SARS-CoV-2, which of course, requires a very high level safety training for use, which we have at Sinai, but a bit of an easier way to measure it that's accepted in the field is using something called pseudoviruses. So what's really interesting is that you can use a harmless virus. You can engineer a harmless virus to carry whatever surface protein you want. And there were already systems in place, mostly developed because of HIV research originally, that use pseudotyped viruses. So in this case, I use a virus called the vesicular stomatitis virus, or VSV. VSV is essentially a harmless virus, and it's been engineered. And this is from a collaborator lab of ours who made this system. Many labs already had systems like this to study SARS and MERS, so other human coronaviruses. So it was an easy switch to sub in the SARS-CoV-2 spike. So we use a VSV-based system, which is all VSV virus, but on the surface, it actually has the SARS-CoV-2 spike protein. And we incubate those VSV viruses that have the spike on their surface. We incubate those with the antibodies that we extract from the milk. And then we add cells. Those cells have ACE2. Okay. And the system is also very useful because the viruses were also engineered to make the cells glow green if they were infected. And it's not a visual green that I can see, but it's, again, what a machine can detect as fluorescence. So then you can measure that output and the reduction of that green output by the antibodies from the milk. So this is how we measure neutralization. So I'm just going to give you a very quick snapshot of just the bit of data that we've acquired looking at neutralizing antibodies. And essentially the take-home message right now is that all the extracted milk IgA that we have studied, which is just a handful of samples right now, they exhibit potent neutralization against this spike pseudotyped VSV. So what you can see here is I've looked at 5 samples here. We've looked at a few more than this, but this was essentially the first 5. And all of them reproduce this. So you can see here that these 5 samples, which we did start with sort of those more potent responders, and then we're going to work through the samples. And what you can see is that this extracted IgA has at a certain concentration, we use a relevant concentration that might be found in milk. You can see that there's essentially 100% neutralization at that particular concentration, and you can dilute this out as well. And then you can compare to the negative control samples as well, and we see, in this case, this is looking at 5 pre-pandemic controls. You have very little background activity that truly neutralizes the virus. So this is extremely promising. And of course, we are working through looking at more samples right now, looking at the 2, samples from particular people, the longitudinal aspect. And then we will also be looking at live SARS-CoV-2 virus in the really near future to compare to this pseudovirus assay. So that was definitely a lot of information, and I think I've probably summarized a lot of it already, and just to work through it. And then I'm really, really happy to spend a while. I'm a little bit early, so I'm happy to spend a long time answering a bunch of questions. So just to summarize, so again, 95% of milk samples from COVID-19 recovered participants were positive for secretory IgA specific to the virus. And again, as I mentioned, this is what we call just a snapshot of this immune response. Immunity is dynamic, so it changes every day, it changes over time, it changes in response to different stimuli, and that's why this study is longitudinal. So we are looking every month ultimately at all of these samples, and especially interested in the first and last samples we've gotten from people, which is, I showed you some of that data. So a much larger sample size than what I showed you and long-term follow-up is really needed to really understand this effect and if it's truly protective for babies. And that is what we're doing right now. So it's quite a big study. As I mentioned, of course, for obvious reasons, this is really important for public health and public policy. And so far what we found, which is really important, is that these antibodies are durable. So they're lasting over time, and they're neutralizing, the ones we've looked at so far. And so, as I've mentioned a lot, because I'm really interested in this, it's unprecedented, but the secretory IgA can be extracted for use as a highly targeted respiratory therapeutic. That's how I see it, which is different than convalescent plasma and convalescent plasma antibody, which is given intravenously and is mostly IgG and is unlikely to survive as well in a targeted way. Although that has not really, I'm not saying that's been really tested or compared to milk antibody, but that's something we really need to do and have started doing. We're starting off in our small animal study using extracted milk antibody. We're going to be doing that in the next 6 months or so, extracting the milk antibody from probably our highest donors, highest antibody levels from many donors, extracting that and using those to see if it can prevent infection. In this case, we're going to use hamsters. Hamsters are a very good model for SARS-CoV-2, believe it or not. They can infect each other through droplets just like us, and their pathology of the disease is a pretty good model for human disease. So that's what we'll be using, and we're going to be testing to see if these antibodies are truly protective when given therapeutically. And as I said, secretory IG is really durable. So that's why we're so interested in it to use as a therapeutic in the nose or the upper respiratory tract. And, if you know milk and you know milk donors and really understand milk production, it can be, milk production can be increased once it's established. Everybody wants to help this pandemic. You can give milk every day. We can get a really efficient extraction of antibody from milk. And I believe that, if there's interest in this as a therapeutic, with a strong global recruitment campaign, I believe that sourcing this milk for the therapy would actually not be, an obstacle. And I think it could be a really applicable therapy because even though there's vaccines now, there's vaccines here. But globally, this pandemic is not going away on the same timeline as I really hope it goes away, where I live, unfortunately. So I think therapies like this might still really be really important. And it goes beyond SARS-CoV-2 and COVID-19, because if we have the interest now, because milk is getting attention now, milk immunology, and we can prove a principle that you can do this therapeutically, then it can apply to all sorts of therapies, right? And we are learning so much about milk immunology from this that can be applied to all sorts of other diseases and protecting babies from other diseases as well. Studies that just have not been done because milk is so understudied. So there's an importance here well beyond the pandemic. And so that's the end of my talk. And I just would love to thank, everyone here at Mount Sinai, especially Alyssa Fox, who's my technician, who really has done, almost just all of what you've seen today. The collaborator labs who've given us proteins for our experiments and the use of their neutralization assay. I also had a lot of really generous research coordinator and materials support for the pilot studies from UC Merced. And I also have support, donor support from Medela, which has paid, especially in the pilot studies, for milk shipping via Milk Stork. I'm supported by the National Institute of Allergy and Infectious Diseases for this work. And of course, none of this could be done without the generous milk donors. So that's it. And as I said, I'm really, really happy to take questions. Thank you.
Wow, thank you so much, Dr. Powell. This was just such a wonderful presentation. It was so informative and just exciting, what you guys are discovering. And I feel so lucky that you guys are here in New York City, and we are fellow Brooklyn parents, and it's just really exciting, everything that's happening. So we do have a lot of questions, and so I think I'm just going to read the questions as opposed to having the asker read them, just in the interest of time, and I'll try to make sure we're touching on everything and there aren't any repeats. But the first question is, I worry about the ethics of breast milk therapeutics and the potential for exploitation of vulnerable mother-parent-infant dyads. Do you have any thoughts for protecting donors from exploitation by pharmaceutical companies?
Yeah, that's definitely crossed my mind. And I think, for my own studies, obviously, when there's human research, there's a really significant consenting process and making sure that nobody is being exploited. And I do offer, monetary compensation for these studies, which would I certainly, if I were in charge of, if this goes in the direction I'd like it to go, certainly monetary compensation would be part of it, which is always optional. And I think, especially if you're talking about recruiting donors globally, I would really, want to make sure that the, IRB protections that, that are given to my participants here, in the USA would be extended over, would have supervision over any, recruitment that would be going on globally. So yeah, I do think about that, and we'd have to really brainstorm and make sure nothing, bad happened in that sense.
Yeah, that was a great question. Thank you. Okay, so there are a lot of questions about the vaccine and lactating parents taking the vaccine. A lot of them comparing and contrasting the response between actually having the infection or receiving the immunization. So is it expected that antibodies created by a vaccine might behave similarly to those created by infection? Could it be beneficial for children who are not currently breastfeeding to receive breast milk from a vaccinated mother?
Yeah, so that's a really great question. And we have, as quickly as we can, and this kind of felt similar to how we were feeling in April with the rush of it all, to get the data out there, even before we were funded. We have initiated a vaccine side of these studies, of course, because it's so important. So that we've already had a big recruitment for that. Essentially all, healthcare professionals that were in the first tier of people getting the vaccine. And we should, I expect that actually there will be differences in the antibody response. I don't know that those differences are going to be a bad thing, but there's there's some data using non-human primates. So that means like higher order monkeys, where some experiments are done for immunization and looking at milk production and antibodies in there, and that they've been done in the HIV field. And we know from those studies that often vaccination does not lead to the same response as natural infection. And there's it's really, really understudied, so understudied that we don't even know. So it's very exploratory. So in the next couple of weeks, I'm going to have just the really early data to try and start comparing to infection. And again, it'll be a similar study, a long-term study, looking at people over time and their antibody response. So that's super important. And again, there's so many different vaccines that are going to come down the line. So they could each have a different response. And that's a really big deal, because one day when you can choose which one you want, if that happens, there might be the one you should get, so you could be protecting your baby. And so, we need to know this information.
Okay, that's great. Thank you. So you did touch upon how you're still seeing antibody response in the human milk like months after the parent recovers.
Yeah.
Is there any reason to believe that antibody presence in the breast milk will persist even longer after the lactating individual recovers? Like how long are you seeing that?
Yeah, so in a lot of the samples I showed in that figure, most, well, as I said, they're all, there's a couple that are 5 months apart, a couple 6 months, and a lot of them are actually 8 or 9 months apart. So about as far apart as we could have at this point. Wow. And there's no change, and that's the thing. I was shocked to find out as an immunologist when I got into this area, and why I got into it. Of course, it was from personal interest, being a breastfeeding mom and wondering like about the science of it all, and realizing nobody studied anything really, relatively speaking. So, you might want to ask, hey, I had, insert whatever, infection. Is my baby, is the milk, going to probably protect my baby, 9 months after? Nobody knows, not against SARS-CoV-2, not against anything. And that's shocking, right? Just shocks that no one has really studied that much.
Right.
So that's insane to me. And so, I didn't know what we were going to find, because the data from blood has been kind of, at first it was a bit all over the place. Originally people were saying, oh, the antibody levels in serum dropped a bunch after 3 months. Now, finally, with really good studies, we are seeing that although there is a drop, which you would expect, it's very likely that, based on blood, people are protected for at least, 6 to 8 months after they've been infected. That's what very recent, really good studies are showing of, over 1,000 people. And so in milk, what we're finding is, certainly not, I don't even see the drop. That's the thing. I'm not,
Wow.
I haven't really found evidence yet, in the samples we've looked at, that there's much of a notable drop at all. And I don't know what that means, and I can't compare it to any other infection because the data is not really available. But I think milk is really special. I think the antibodies and the immune response in milk is really special evolutionarily, and the way it persists, actually. So I think that's what we're going to find.
And were you able to measure any of the the blood plasma levels of any of your milk donors to compare the activity in the plasma as compared to the breast milk?
Right. So we didn't do that for well, yeah, I don't think any of the samples I showed you, but it is a sub study. So I will ultimately have data for a subset because I'm interested in that. And I expect there's going to be some differences which may feed into a larger study, comparing and looking over time. So yeah, so we are doing that.
Okay. We're having a lot of questions of how people can participate. And I did, so at the top of the chat is your email for the study. So people can email your team through that email address. I am also putting it in our survey information. So if you don't catch it there, you'll catch it otherwise. But is that, I know you said you're looking for local, like New York City, folks who've gotten the vaccination and are lactating.
Right. Yeah. So we've had, as I said, really a massive recruitment. So if you email, you'll see in the auto reply that currently we're only open to NYC, like 5 boroughs, participants who are getting vaccinated, like, soon now, like they're prioritized, or they had recent COVID-19 infection. We're open nationally to anyone with recent, like a month ago or less COVID-19 infection. We're still open for that nationally. And then in terms of additional national enrollment, if you are in a trial for a vaccine other than Pfizer and Moderna, and you're out there somewhere, we are recruiting nationally for people who are getting like the J&J vaccine or anything else that's coming down the line, because the J&J vaccine trial does not exclude lactating people. I know that.
Oh, okay.
So there are people I'm trying to identify to get into my study who have got the J&J vaccine. And so, yeah, so that's still a national. So if you know anyone, pass the word. Yeah, if you email, you will see that auto reply, just so you know in advance that outside NYC, we're closed to a lot of enrollment right now.
And I did want to highlight, you did talk about this a little bit, but at the beginning, at least, you were like on the subway, going to people's houses and picking it up yourself. That's amazing.
Yeah, well, that, it's sort of what happens when you start doing studies in a panic, when you don't have the money to pay anybody else to do anything for you. So yeah, that was what I was doing really in April and May, was taking the train, taking the bus. And I mean, nobody was really on it, so it felt okay. Obviously masks and everything. And I was picking up these milk samples, bringing them in and all of that. So, but it wasn't just me. I mean, obviously I had people in the lab doing a lot of the work coming in, and they were taking the train to come to work too during the pause, so.
Right.
Yeah.
Okay, let's see. Did you notice any color changes in the COVID positive breast milk samples?
No, I didn't. I mean, when you collect milk, all the samples just look different all the time. That's the thing, so specifically, I haven't really taken, we didn't really visually look at like 100 COVID samples and visually compare to like 100 non-COVID in any meaningful way. It's all anecdotal,
Right.
but yeah, I mean, when you collect milk, you see a lot of different kinds of milk, like more opaque milk, more translucent milk, milk with weird hues of color. But yeah, I mean, associating it with COVID-19 infection. And remember, this milk is not was not collected during acute infection, right? So this is not, we deliberately did not collect the milk when people were contagious for obvious reasons. And especially because we didn't know at the time if that milk would be infectious to us in the lab. Right? Which now all the data really, I would say definitively has shown that milk itself is not a vector of infection for SARS-CoV-2. I was not personally looking at that, but we didn't know. So, and also, I mean, just the handling, even if the milk is not, but just the people handling it or breathing on it, or, it's in their homes. So we, protected ourselves by making sure that we actually only picked up milk, at least 3 or 4 weeks after they had been in, had symptoms essentially. Yeah.
Okay, great. That's great information. This is a great question, Margot. Is it possible to replicate the SIGA in a lab? So once you've taken it from the milk, is that what you can do that as far as creating a therapeutic, like you were talking about?
Right. So yeah, so that comes up, why bother, getting it from milk? Or I think a lot of people who actually don't really understand much about milk or milk production come at me and say, well, you'll never get enough from milk anyway. Let's just make it, in a lab. So the answer is yes. You can make secretory IgA recombinantly. That's what we call it. It's not easy. Okay. So making recombinant IgG, which is like the therapies that like Regeneron and other companies have made that you've heard of, that are like IV infusions of antibody, those are IgG, because that's just the standard. That's what you find in blood. That is the standard easy thing to make. Anything that's a monomer, IgA as a monomer, whatever, meaning like a single antibody molecule, is pretty easy to make these days. Secretory IgA is not easy to make. So we have struggled with it personally. We've done it. It's hard to make it in high amounts because it's so many different proteins, right? So it's a dimer structure. It's got another protein holding them together, and it's got the secretory component protein, which you have to have being made by the cells, wrapping it all together, and then you have to purify that giant molecule. It's not an easy thing. Now, companies, that are more sophisticated than what I've got going on here, I'm sure are doing it in some level, but it's not something that's on like a massive scale that has been happening, So it's almost like, to me, like the milk is more like, I don't know, like the grassroots, let proof of principle, if we can show it in milk, if we can do it with milk, then that will push the research to say, hey, let's do it, let's make this in a lab, with these consistent batches of whatever we're going to make. So you can, but it's hard. That's my answer.
So essentially we need to probably be taking it from milk. That's what you were talking about, like on a global scale, collecting milk ideally, and creating a therapeutic for parts of the world that don't have the same access maybe to the vaccines, or where it hasn't died down, like hopefully it will here.
Yeah, I mean, it certainly hasn't died down here.
Right.
There's still a lot of room. There will still be a lot of room for therapeutics here for a while.
Yeah.
And I think as a therapeutic, it could be alongside other therapeutics that are already on the market or being tested. But yeah, I mean, I think I can see a reasoning for it for a long time outside of, high income countries. Yeah.
Okay. Okay. So a question was asked, or did Dr. Powell collaborate with others performing research internationally?
So for these studies, none of it, I mean, it was not from any international collaborations. I have a collaboration going in the UK to share samples from there, test them similarly, and also so we can look at samples from the AstraZeneca Oxford vaccine, because that's not licensed here, but it's licensed there. So we're going to be doing that. And I don't have other international collaborations going, but I mean, I would like to just to access milk samples from other licensed vaccines that I can't otherwise get a hold of.
Yeah. Okay, that's good to know. And then there was a question, where can the published studies be found?
Yeah, so our published study on the pilot data is in iScience. So that's a Cell journal. So you can go to just search Cell iScience, my last name. I think it's publicly available. I think it's like free access at this point.
Okay.
But if anyone's really interested, I can send around a PDF of the paper.
Yeah,
if
people
there's
are really
request for
interested.
that. Yeah, I think it's free online, but if it's not, let me know.
Okay. All right. Okay. Oh, okay. This is an interesting question from Janine. Does the person infected with COVID need to be breastfeeding at the time of infection to have antibodies? Would a pregnant or pre-pregnant person who had COVID produce breast milk a few months later that protected their baby?
Right. So most of these people in the data I showed you got infected once lactation was established with that current, baby. But about 3 of them were infected during pregnancy, actually. And certainly, I mean, it's a small sample, but their response was no different.
Okay.
I know that. It is something we'll look into a little more as our sample number grows and we differentiate more between groups.
Mhm.
We'll compare people who were infected, during the 3rd trimester or earlier, because at first we didn't have, anyone who would have been infected earlier than like the 3rd trimester. But now that time has passed,
Right.
for better or for worse, we will have samples like that, right? And similarly for the vaccine studies, that's a question too, right? Like, how long does it last? What if you got it vaccinated during your pregnancy? Is there a difference between early and late pregnancy? I mean, again, the data even for other viruses is not really well established. So it's kind of a black box. But yeah, I mean, from what we know so far, I wouldn't say there's been any difference.
Okay. And have you noticed any, this comes up a lot in professional circles, but any supply changes if a parent who has an established milk supply, they get COVID, have you seen their supply be affected?
Not that I know of. I mean, we don't ask that. No
Yeah.
one's really volunteered that kind of info. I think it would, my guess would it's not, if it's something, it's just because, you're sick. So that can affect supply no matter what.
Right.
I think, one aspect of that is that mothers and babies were being separated a lot, especially early on. So sometimes establishing breastfeeding was really hard because of that. And then sometimes it messed with the supply because they were separated for, a couple weeks. And I definitely have women in the study who were separated from their babies.
Right.
During the early days, which is definitely still happening all over the world, even though all the data says don't do that.
Right. Right, especially with what we're finding from your work, that it's so important that the babies receive the milk, and the best way to do that is to be with their parent.
Right.
Yes. Okay. What can we? Oh, okay. So you did kind of cover that you predict there will be a difference in antibody response if you have the infection versus you're vaccinated. Okay. So that was good. Okay. When you are doing, this is a great question, Carmen, when you're doing the tests on a parent who's been vaccinated, do they have to be fully vaccinated? Are you looking at initial response with just one vaccine and then the second vaccine?
Yeah, I mean, we're looking at everything. So, my super early data that I'm going to have in a week or two is looking for those people probably about 7 days after the second shot.
Okay.
So, by about 7 days in the blood, the protective effect is there from the efficacy studies, right, from the trials. So that's, really the earliest I want to look right now, and then we'll sort of work backwards. Did you get a response after the first shot? and how common was that? Was it different? And then, we're also going to have people in the study. So then that's for different vaccines, obviously. And then, the 14 days is an important time point, 2 weeks after the second shot. Then we're going to look monthly, similar to how we're doing the infection study. And then, we're going to have a subset of participants who actually were infected with COVID-19 previously, and now they are getting vaccinated. And that, I don't know what's going to happen.
Yeah, that would be
How it
fascinating.
will, I guess, modulate the response or not,
Mhm.
how it will boost it. Yeah, so it's very complicated because now we have all these different groups to study and
Yeah.
compare to each other. Yeah.
It's a lot to keep track of. Oh, and thank you so much, Tova. Tova Ovitz posted in the chat for anyone looking for the link to the research in the cell, the iScience link is in the chat. So go ahead and copy that if you haven't already. Is there a difference in the immune response to SARS-CoV-2 in the event where the infected person is the mother versus the newborn? I guess, did you have any who it was just the newborn who was affected?
Yeah, not to my knowledge. So, that's just not how we recruit, right? I mean, you have to have had a positive COVID test yourself. And then we do have symptom data on these people, and whether their babies were, noticeably sick, tested positive or otherwise. But yeah, we don't recruit by like, was your baby sick, but what, so I don't know, we won't really have the answer, I guess, to that. But in all things, if let's say the baby was the first to infect the rest of the family, which is what I would expect. I mean, I don't know how the baby, the baby's pretty likely to infect the mom,
Right.
with the close contact and not knowing. The baby would have the initial an immune response that came first, so to speak. And there is interplay between the baby's mouth, the breast, exchange of information, so to speak. We haven't looked at that at all, but I'd expect we'd see something interesting in terms of a local response in the breast. But the mom is going to have to get infected to really have this robust immune response, even if there's some local interaction between what's going on in like the baby's saliva versus the breast, local breast immune response. But that's not going to cause this robust response in the mom that we're measuring. She would have to get infected.
Yeah, that makes sense. Okay. Do you have a minimum time from exposure to first detected IgA in the milk?
Yeah, so we are sort of going back and trying to look a little earlier. Like I said, we didn't do that to protect ourselves because we didn't know. And we still have to be careful just because of the sample handling and everything. But everything's a little better understood now than it was in April here. So yeah, so for some of these people, we do have some earlier samples we haven't looked at that we kind of left alone. And then for newer recruits, we have samples basically like right from infection. But another group looked at early samples because they were looking for virus to see if it was there. A group out of Idaho and Shelley McGuire's group, and I know that they found the IgA response came up really quite quickly. I don't know exactly the day.
Right.
And so, we're going to look at our samples a few different ways to also see what we see in, a larger group and looking at the different classes and, of antibody and seeing how quickly you get that. But I do expect, it is fast.
Yeah.
Faster than what might be detectable in serum.
Right. Okay. What happens to the antibodies once the baby receives them?
Yeah, so that's a good question. Like I said, these antibodies evolved to be durable, right? And there's a reason for that, which is because they're acting, in various mucosal environments. So the infant mouth, obviously, the infant, nasal cavity as well, because we've all watched babies breastfeed, and there's a lot of interplay there going up into the nasal cavity. And then, once you're in the nasal cavity, I think that there's probably, I would say it's, the you're going to get a lot of activity in that sort of upper respiratory tract because of the, the milk going up into the nasal cavity to a certain extent. I think a lot of the action is really in, that oral environment. There is action that's going to happen in the upper digestive tract. Absolutely, because a lot of the antibodies survives that upper digestive tract and even the stomach. And there's studies that have been done to show that. So I think especially with SARS-CoV-2, there is a lot of GI involvement in the pathology of this illness, unlike maybe other respiratory type infections. And that's the funny thing about COVID-19. It's a respiratory infection, and ultimately we associate so much to do with the respiratory system, with the severity of the disease and being breathless and everything, of course. But, ACE2 is expressed on so many different cell types, and it's really, it's literally on our blood vessels, this receptor, right? So it affects all sorts of systems. And I think that, you have to think about where the antibodies are going to do their action right away, and so if the baby's being exposed to the virus, they're getting exposed through the nose and mouth, just like the rest of us. So that, I think it's more of an immediate, action. I'm not saying that the antibodies are somehow in the lung, necessarily. I don't think they are. It's hard to imagine that mechanism, at least in high quantities, but they're in the gut, they're going down. So, again, if virus is replicating in the gut, which it likely is in some people, that there would definitely be antibodies there to stop that,
Okay. Wow. So Teresa Landau has two great questions. Has your study been replicated by other researchers in other labs? And she says, fabulous work. Thank you so much. And then also, is there a role for milk banks?
Yeah, so definitely, if you just do like a literature search on the topic, you can see that we're not the only ones who are studying this. There are various other researchers in this country and in other countries. I think we have the biggest study, probably just because of the local NYC, moment where we started. And, because it hit us so fast and so hard, really early. But I'm not the only one who has found, this effect at all. So, you can see that in the literature. Milk banks. So yeah, I mean, that's interesting. We actually have a collaboration with a company. It's a Canadian company called Lactiga. And they have a really good method to extract milk antibody. And so we're using that method, and they were actually into using milk antibody therapeutically before COVID-19. That was sort of the premise of their company, for using it for treating immunodeficiencies in general, especially like as they affect the gastrointestinal tract. And they certainly have targeted milk banks as part of our collaboration, with the idea of sourcing milk from milk banks, especially because actually you're excluded, the milk donors are excluded from donation if they had passed SARS-CoV-2, or they were at a certain point. I don't know what's going on now, they changed the regulations, but so that, it's mostly like discarded milk, essentially. So there's definitely a role for milk banks in all of this, for sure.
I mean, we're so lucky here in New York to have the New York Milk Bank. It would be great if you guys could collaborate in some way.
Yeah, I mean, certainly for the therapeutic aspect, yeah, I mean, there's definitely a place for that, for sure.
And and Roseanne just asked, actually, and she's part of the New York Milk Bank. We're often asked by our milk donors if receiving the COVID vaccine would prevent them from donating their milk. And she says that we do not defer for having COVID or the vaccine. Roseanne, is that true? Just wanted to confirm that. But, sometimes there are things that preclude you from being able to donate. So, we do have a question. Are you keeping track of different data from the donors as far as race or ethnicity? Someone wanted to know if there are minorities that have been donating milk to the study.
Right. So officially, that is not a question in any of my surveys, but I mean, I know just because, especially with the New York City enrollment and this being, a very diverse place, that it is, the diversity is pretty high in the study, just because of the NYC involvement especially. But I don't have that information. I don't really ask it.
Okay. Okay, great. Okay. Can you address the stability of the antibodies when you're reheating or preparing expressed milk for the baby to, eat in a bottle?
Yeah, so I haven't done this work, but I know that there's recent work that shows that essentially the pasteurization type treatment that's done, at milk banks, the antibodies are okay. I don't know that they're 100% there, but it kills viruses. That's why we pasteurize. That kind of like quick heat treatment does not kill the antibodies, so to speak. That's what researchers have shown. So that's a good thing. I can't speak to each individual, how they're heating and what temperature, what they're doing. I wouldn't be like boiling anything for long, but I mean, what we do here in the lab, we heat and activate samples, which means that we do about 60 degrees Celsius for 30 minutes. So that kills viruses, while protecting antibodies. And it also destroys other immune factors that would mess with our experiments that are not, they're not particularly important in this context.
So then a parent who's just warming some milk,
that's fine.
In the fridge, that's fine.
Oh yeah, normal, again, I mean, repeated freeze thaws, it's not advised you do that really anyway.
Right, no.
That's not good for proteins. But just warming up under the stream of hot water or, mixing around, I wouldn't worry about the antibodies for that.
Okay, great. How much milk do you think would be needed for a therapeutic response? So for example, how many people could receive SIGA from a single ounce of milk?
Yeah, I've done some like back of the notebook calculations, I think, I don't have them in front of me, but it's essentially, we don't know the dose that would be required therapeutically. That's number one, right? I mean, we can estimate based on some vaguely similar other studies that don't use secretory IgA. But we are doing those animal studies to get a sense of that. So there will likely be a dosing aspect either in the first study or in a follow-up study, to better understand the dose that is required. And then you can kind of extrapolate to people from the small animals. But, I've done some calculations and, there's about half a milligram of IgA per milliliter of milk. And, you can give milk every day. I think donors, were they so incentivized and eager, I think that you could easily get, a few hundred milliliters from each person, every single day. And that really adds up. You can, you can get out the calculator yourself. But I mean, if you're talking about even just 1,000 donors, which I'm talking about global recruitment, I'm not even, 1,000's a very small number in that context. But, I think it's very doable. But the precise numbers, I can't tell you because we just don't know those doses. But I do expect, this is a really targeted therapy. So I think that when you can't compare the amount of antibody that's used by intravenous therapy when you're talking about the monoclonal antibodies or plasma IgG from blood, it's not comparable. So those do use very high doses, which you could not achieve with what I'm talking about. But I don't think that's relevant because I'm talking about like a targeted respiratory therapy that's highly durable.
Okay. Wow. So we do have a bunch of other questions, but unfortunately it's now 1:30 and you've been so generous with your time. I want to be respectful of everyone's time. So I'm going to wrap things up now. The last question is from one of our NYLCA members who asked, what can we do as IBCLCs to best help you with this important research?
I think just keep spreading the word, really, not so much for enrollment, but just, that you have seen this data firsthand, so to speak, of how the antibodies are there and they're durable, and you can't speak to every individual case, but you probably get a lot of questions about, should I be breastfeeding? What if I had COVID 9 months ago? Should, all of that. And I think just spreading the word and informing, your clients and also doctors, nurses. I don't know who's listening today, but I think that's always a big gap, people who don't have the right information, and you can direct them at the, at the published data, and there's other published data as well, not just mine. Yeah, so I think that's really just the most helpful at this point, for sure.
And we've been recording this, and we're going to put it up on our website and our YouTube page. So, you can forward the link to anyone. So this information, it's really important that this information is available. So you can share it with all of the pediatricians you collaborate with, and make sure that they know that it's okay for a parent who, has tested positive to be in contact with their baby and feed their baby. It's actually like essential that their baby receives the milk. So, I think that's it for now. I'm going to see if I can copy some of the questions, and maybe I'll be able to reach out to you just for quick answers. But otherwise, as we're wrapping up, I just want to let people know that when this webinar ends, there will be a pop-up. So make sure your pop-ups are enabled for a survey. We appreciate any feedback you can give us about this presentation. And then also, if you're looking for SERPs after you complete the survey, there'll be another link to the SERP survey, so we can collect your information so you can get credit. Credit is only for those, SERPs are only available for those who participated in the live presentation, because we can verify you were here. So thank you so much, Dr. Powell. It's such a pleasure to talk to you and to connect with you in person. And this research is obviously so important and essential to the work that we're doing. And we as IBCLCs and lactation professionals will do our best to make sure that we keep families breastfeeding and chest feeding, and that human milk is available for everyone who wants it. And we're really encouraged, maybe we can bring you back, and when you have more results, to talk about, the latest things. It sounds like you have amazing plans upcoming as well.
Yeah, thank you so much for having me.
Crystal, if you wanted to say anything, go ahead.
Yes, thank you, Dr. Powell. That was really, really informational. The Mount Sinai team of lactation, we are here and ready to spread the word. And if you need anything from us, as one of my colleagues.
Yeah.
Thank you very much.
Awesome. And we're just very excited that we knew this was possible, and we're just glad that you're able to, verify that information for us. Thank you so much.
Yeah.
Thank you so much. Be well, be safe, and we look forward to all of the research coming up.
All right. Thank you.
Thanks, everyone.
Bye. Thank you.
Bye-bye.